Morning. I'm Alex Albanak. I'm the Associate Vice Provost for Research Administration at Johns Hopkins and FDP's co-chair. On behalf of Michelle Masucci, my fellow co-chair, and David Wright, the Executive Director of FDP, I would like to welcome you to our third online meeting. We're very pleased to see so many people that have joined us in our virtual meeting format. Although we look forward to reuniting with you, hopefully soon, but in the meantime, we have over 750 people registered for this meeting. This meeting is the first in our phase 7 of FDP, and we're welcoming a lot of new institutional members. We have 217 institutions as our members, and this includes research intensive, emerging research, and minority serving institutions. We partner with 10 federal agencies who will later this afternoon will be providing federal updates. One of our most important goals for this phase is to increase the impact of FDP. And this is going to be through broadening our participation of the organization and working together to find ways to improve the quality and efficiency of research administration. In addition, because we have so many new members, throughout this meeting, we will be sure to incorporate program elements that will help to onboard our new members. And also, just in case you missed it, we've hosted 2 virtual onboarding webinars, and they're both posted on the FDP website. And this goes over a little bit of FDP's history and our structure and different areas of focus that we'll be looking at in phase 7. In addition, this meeting will include committee update. This is later in the afternoon. I hope you can all join us. It's going to provide an overview of some of the current initiatives, as well as discussing different opportunities for volunteering. So at this point, I'd like to also mark a milestone of our most recent co-chair, past co-chair, Dick Seligman from Caltech. He's retiring this month. We are also grateful for his leadership and help us prepare for phase 7 and for his dedication to FDP as one of the earliest members and advocates. At this point, I'd like to turn this over to Michelle.
Thank you, Alex. Welcome, everyone. We're really thrilled that you could be with us today. Just a few program notes. I want to remind you that it's not too late to register for the meetings and that all of your colleagues from your institutions are welcome to the sessions that might be of interest to them. As you know, as Alex pointed out, our agenda today includes today's plenary presentation, our federal agency updates and committee updates. But we're also really want to make sure that you're aware that there's a happy hour later on this afternoon. And if you'd like to meet some of the FDP members, and we'd love to welcome some of our new members, that's an opportunity to do so. It's a big collective, and we're not really sure how it's going to go, but just come and join the space and we'll get to know each other a little bit better and more informally. As you may know, the Federal Demonstration Partnership is an initiative of the Government University Industry Research Roundtable of the National Academies, better known as GWIR. And we are the acronym organization, so I'm going to acclimate you right off the top. Our relationship with GWIR is one of the distinguishing features of the FDP because it means that our activities are connected to the important convening role that GWIR and the National Academies play in connecting our work to broad societal discussions, as well as those within government agency, advocates for research, and our member institutions. It's my distinct pleasure. I'm really delighted to introduce you to Susan Sloan, who is the director of GWIR, and she will introduce today's plenary speaker. She has served in the role of being director of GWIR since 2008. Prior to her distinguished tenure at the National Academies, she worked with a number of research organizations and foundations, including the National Science Foundation and the American Association for the Advancement of Science. She also served as the Associate Director of the Master of Health Science in Health Policy program at Johns Hopkins University. Her unique background working at the crossroads of policy funding agencies and the research community, including with faculty, make her a tireless advocate for what we do and an invaluable resource for all of us at the FDP. So with this, I am turning over the session to Susan, who will welcome our plenary speaker. Susan.
Thank you so much, Michelle, and to everybody here today. I too want to welcome everybody to this first official meeting of the FDP Phase 7. and particularly offer a warm welcome to the new member organizations. As you've heard, we're now at 217 institutions. This is up from 157 in phase 6. The FDP serves a unique and vital role in the reduction of administrative burden across the research enterprise, and we want to thank you all for your interest and commitment to the goals of this organization. As you've just heard, my name is Susan Sloan. I direct the Government University Industry Research Roundtable, and GWIR serves as neutral convener to the FDP within the National Academies of Sciences, Engineering, and Medicine. But it is its own membership organization that focuses on science and technology policy matters. Our plenary speaker today has long served and plays a valuable role in the leadership of GWIR, and I'm just truly honored and honored to be able to introduce him this morning for what is bound to be a very compelling discussion. So Dr. Christopher Austin has served as director of the National Center for Advancing Translational Sciences, NCATS, at the National Institute of Health since 2012. Prior to this role, he was NCATS scientific director, focusing on the translating basic science discoveries into new treatments and technologies to improve the efficiency of therapeutic and diagnostic development. He founded several initiatives, including the NIH Chemical Genomics Center, the Therapeutics for Rare and Neglected Diseases program, the Toxicology in the 21st Century program. Before joining NIH in 2002, he led genomic-based target discovery, genomics, and neuropsychiatric drug development programs at Merck. From 2016 to 2018, he served as the chair of the International Rare Disease Research Consortium. Dr. Austin is also a member of the National Academy of Medicine. He earned an AB from Princeton University, an MD from Harvard Medical School, and completed training in internal medicine and neurology at Mass General Hospital. So with that, we're so pleased to have you, Chris, and we will turn the microphone over to you.
Thank you, Susan. You can all hear me, correct? Yeah. Okay, well, let me know if you can. So it's a real pleasure to be here today for all kinds of reasons. First, I want to thank the FDP for having me. I've been privileged to know a lot about the FDP, not only through GWIR, but through my work at NIH, of course. And for many, many years, my co-representative to GWIR was Jody Black, who I think a lot of you know at the NIH and OER. And being a part of GWIR has been just a remarkable experience for me. And those of you who have not been involved in GWIR, the GWIR part of what GWIR does as opposed to the FDP part, I really would urge you to follow what they do perhaps a little more closely and attend some of the meetings. You know, for me, who spends really all of his time, or most of it anyway, in the life science space, one of the things that I've found particularly useful about GWIR, in addition to bringing together government and universities and industry, is the interaction with our colleagues in the physical sciences, the NSF, in engineering, so the National Academy of Engineering, National Academy of Sciences, as well as the National Academy of Medicine, are involved in GWIR, and that's just part of our research enterprise that I just have had less interaction with, and we have a lot in common on a lot of things that we can do together. So keep it in mind. And I have to give a particular shout out to Susan Sloan, who is really responsible for a lot of the successes that GWIR has had, and just so grateful for her leadership. So this talk actually came from a presentation that I gave to GWIR back in October, and I hope you'll find it equally useful. I think one of the takeaway lessons is that though COVID-19 has caused immeasurable, unprecedented suffering and death in this country and around the world, what we've also seen is the rapid adjustment in some very positive ways of our research infrastructure and collaborative infrastructure in ways that I think a lot of us have hoped to see for quite a long time, and they have happened during this last year. A lot of flexibilities that on all sides that I think a lot of us would like to see continue. And one of the things that I'd like you to think about, and we'll come back to at the end, and something which is really weighing on my mind now is, well, how do we, in addition to hopefully banishing this awful pandemic to the history books, how do we capture the good parts and not go back to the bad old days and all the frustrating issues that all of us deal with in trying to work with each other. That we feel your pain every day, and don't like these the complexities and costs any more than you do, and so real question for us as a community is, well, how do we work together to try to maintain some of the good sides? So, okay, so let's just dive right in. Next slide. So I just want to remind you that just to start, I can't start out a talk like this without just showing you what the numbers are. This is, of course, a public health crisis unrivaled in modern times. Not since the flu epidemic in 1917-18 have we seen such a thing. And those numbers have continued to increase, and these are the numbers as of yesterday. And you'll see the numbers of cases, hospitalizations, and deaths continue to reach new highs. So this is something which has occupied me almost completely for the last 9-10 months now, and I know has occupied most of you as well in many professional and personal ways. And so we do need to keep our eye on the ball here to try to deal with this crisis, but also look at well, how have we managed? And I think from my reading, the research enterprise has really risen to the challenge. I mean, what has been done in the last year is absolutely breathtaking. And it's breathtaking because of this trifecta that we talk about: the government, universities, and industry working together in unprecedented ways to improve the health of individuals in the public. And that's why we're all here. And it's happened. Unfortunately, not to the degree that we would like, or else we wouldn't be looking at these numbers. But there have been some positive changes, many positive changes as well. So next one. So I stole a few slides from Tony Fauci, and if you're not used to seeing the Tony Fauci font, you'll get used to it. A lot of bold lettering, and I stole these just because I'm going to talk about a few of these things. So I just wanted to reiterate this: that we know that people are at increased risk for severe COVID-19 illness are older adults, like many of us on the phone, and people of any age with underlying medical conditions. Next one. And these are some of the medical conditions that you go to the next slide, that lead to increase morbidity and mortality: heart conditions, kidney, lung disease, diabetes, obesity being a huge risk factor, cancer, sickle cell disease, immunocompromised, and others. So lots of comorbidities that lead to increased risk. Next one. And we've also seen that this disease has hit racial and ethnic minorities disproportionately hard. This comes from a paper from some investigators, including the director and the director of the National Institute of Minority Health and Health Disparities back in May. But this story has continued, where it has shown us in a very troubling way the disparities which exist in our ability to care for our population. If you go to the next slide, you see these numbers are really quite striking, looking at the differences in hospitalization rates or death rates. They look fairly similar between people of color and white Americans. So something that's addition to, of course, the other social issues which happened last year really have come together to focus us anew on the opportunities and needs in the health disparity space. So next one. Okay, so what did NCATS do? We have a website devoted to this about what our approach was. And if you don't know a lot about NCATS, probably the only thing you really need to know to start out with is that we try to think different in every way. The translational process going from a fundamental discovery to an intervention which is shown to improve the health of individuals and the public, people living in communities, because that's where health happens. That process, I think we all realize, is terribly inefficient and ineffective, and that also was revealed by COVID-19. And so the other thing you might need to know about NCATS is that we're unconventional in the sense that every project we do is a collaboration with somebody. We believe very strongly, and the data point to the fact that translation is an obligatorily team sport. You can't do it on your own, doesn't matter how smart you are, how well funded you are, how experienced you are. You can't do this alone. So collaboration among individuals, sectors, organizations is absolutely critical to make rapid progress. And we also realize that collaboration requires openness, unprecedented openness when it comes to data and resources. And so we took really an unprecedentedly open and collaboration-based approach to COVID-19 because we believed that this was the most rapid route to progress. And I think you'll find that indeed that has turned out to be the case. So next one. So I'm going to give you just a few examples, and probably more than you may want now, but a few examples of what we have done. So starting on the preclinical side, this was an online data resource that we launched in May to quickly share COVID-19 drug screening and drug discovery data. NCATS, as well as other organizations around the world, started screening approved drugs, but those data were not shared in any rapid or complete way. And by complete, I mean the positives and the negatives, all the data on the assays, detailed protocols, and all of that made available within a week of the experiment being done. This has never been done in this translational domain, looking at drug screening and drug repurposing, but the public health pandemic required it, and so we've done that on our website here, and you can see how many visitors and views we've had since then, and we've had really gratifyingly a number of external partners who have deposited their data as well from around the world. Interestingly, only one U.S. academic organization, the University of Michigan. All others, for one reason or another, decided to keep their data to themselves, which I think is a loss, frankly. And we've been shown up by some other countries and a company in Utah. So next one. This is what the Open Data Portal looks like, and I just want to use this this slide to indicate that when we put this together, it quickly came to the attention of the public-private partnership known as ACTIV, or Accelerating COVID-19 Therapeutic Interventions, and vaccine, and the preclinical working group, which I'll tell you about at the end, was looking for a place to gather and publicize data on animal models, large and small animal models for COVID-19 as they were developed. and this was the only database which allowed, or the only data portal which allowed them to do that. So it was used by ACTIV, and still is, to make those animal model data available as well. And you could see these are things that you'd want to know, and all of us would want to know. So for various species, are they good for evaluating vaccines or antivirals or antibodies or what have you? What kind of disease manifestations do they have, and is it severe disease or not? Something that sometimes has been done, but has been done over the course of years, and this all happened within the course of months or even weeks. So next one. We also put together in a different collaboration, this one with the Food and Drug Administration, a mobile app to capture clinicians' experiences of using novel drugs, particularly early in the pandemic. I'm sure all of you remember the almost panic that was happening at the time when large numbers of patients were coming into the hospital, this unknown disease, and disease which hadn't been seen before, and we really had no idea how to treat it, and so there were various drugs that were being used, of course off label in most cases, and again, those data really needed to be shared, not by researchers but by clinicians in hospitals around the world taking care of these patients, and the only reason they were able to share their data in real time on their mobile phone was via this app. And the data has been used, of course, for hypothesis generation, as well as in some cases integration into clinical practice. And it was developed collaboratively, like everything NCATS does, by not only with us and FDA, but also with WHO and the Infectious Disease Society of America. And if you go to the next one, you'll see that it caught the attention of the Critical Path Initiative. The next slide, you'll see, oh, sorry, this just shows you outcome by drug. If you look on the right side, you'll see a lot of the drugs that were used early on and the numbers. And this has caught the attention of a number of organizations, including the ones you can see there on the left, Hopkins, Mayo, et cetera. But then next one, next slide, caught the attention of the Critical Path Institute, and they have put a lot of resources and money into this to expand the purview of this mobile app. So next one. Another thing that we started very early in a collaborative effort with a number of academic institutions and other institutes at the NIH, in this case with NIID, NIBIB, and NCI, we realized that we needed to understand the antibody responses to patients who had been exposed to COVID-19 without knowing it. So we quickly enrolled 10,000 participants all over the country in a population representative fashion. And we did that with the efforts of the University of Alabama and the University of Pittsburgh, CTSAs, and that initial result is going to be published shortly, and then we're going to be following up these patients at 4 and 8 months to see how long these last. And keep in mind, this survey, if you imagine the enormity of this project, 4 institutes enrolling 10,000 participants of the over 400,000 who had volunteered, and to do that through 2 academic institutions and all kinds of agreements and data sharing and all of this stuff, all of this happened within about 2 months. And I'll keep coming back to this. This shows us what the system is capable of. If people are motivated, this can happen. Things happen much more quickly. And I would say the flexibility was on every part. All the institutes, the academic institutions, when industry was involved, wasn't involved in this one, but same kind of flexibility. So next one. Another example is a very large one, is something called the National COVID Cohort Collaborative, or N3C. This is an unprecedented collaboration among virtually all of the CTSA program hubs and many of the Idea State CTR programs and the CTSA Center for Data to Health to create what we all wanted when this started. That is a compendium of real world data from electronic health records across the world, across the country, people who were being seen and treated for COVID, and what their race and ethnicity are, what their comorbidities are, what drugs were being used, and what factors led to more severe disease, and what factors led these patients to getting better. We had no idea how to do that because we had no platform to do it. And in record time, about 1,000 people, and that is the number, 1,000 people from over 100 academic institutions and the IT and informatics group and NCATS, who had been working together quite closely, of course, through the CTSA program, but came together and got this this collaborative, this platform built in about 2 months, and then began ingesting data from institutions all over the country. And these are the current numbers, and they are quite extraordinary, where there are over 2.7 million patients' records in this data set from all across the country, from currently about 40 academic health centers, and of those, over 400,000 are COVID patients. And we have data going back 2 years on these patients, and going forward every week, the data is being ingested again. And you can see the truly mind-boggling amount of data, 2.6 billion rows of data here. So this required, I don't need to tell all of you, the amount of work required to rapidly develop and execute the data agreements, dealing with the federal policy implications, the data sharing implications, the privacy and security implications, which are paramount here. All of this was done with, probably many of you were involved in this, and the formation and operation of a data access committee to determine who can get access to the data. All of that was done in time, which is about 1/10 of what it would normally take if this could ever be done at all. But now just imagine this has been done. So wouldn't this be nice? Wouldn't it be wonderful, beyond nice, to have this kind of data set on coronary heart disease or diabetes or any rare disease you might mention? There is nothing technically preventing us from realizing that dream. Now that we've shown ourselves we can do it in COVID-19, there's no reason it can't be done more broadly. And there are almost 100 now investigators at academic institutions all over the country who have applied and gotten access to the data and are doing their research on these data. So it's opened up an entirely new avenue for academic research. So next one. So I just want to give you a sense of what makes this unique. Some is the numbers, and you can see them in the upper right corner there, but the scale and scope is really incredible. If you think of the data that are coming from your institutions, demographic, symptoms, lab tests, procedures, imaging, medications, lab conditions, et cetera. We've for you cognoscenti, we've taken data from multiple data models and harmonized them. We did all of that work so that the institutions didn't have to harmonize to our standard. We harmonized to theirs, built an internal collaborative analytics platform where teams can work within this platform. And this is, it remains in, the data are held in a secure FedRAMP, if you know what that is, certified cloud, with standardized assessments and continuous monitoring for hacking and other unpleasant things. But I want you to look at the demographics. You may not be able to see this, but you'll see that if you squint, you can see that the age distribution matches the US population. The race distribution matches the US population, race and ethnicity, gender mix, et cetera. And so it really has all the characteristics that one would want in this kind of data set. And again, I want to stress, the only reason this happened is that your institutions made it happen. I mean, you all worked very, very closely with us. I don't want to minimize that, but you all made this happen. Next one. And we can just skip this. This is just technically how it happens. Next one. You're probably familiar with our program called tissue chips, otherwise known as microphysiological systems. These are these microfluidic, multicellular human bioreactors that we've been doing with all of your members for the last 10 years or so. And we rapidly pivoted that effort toward COVID-19 too. It turned out that one of the models which had been developed by Don Ingber and his team at the Wyss and Harvard had been actually a human lung on a chip. And gosh, wouldn't that be useful for looking at pathogenesis and drug response for SARS-CoV-2? And so we were able to do that very rapidly by giving Don and others administrative supplements, some of them through the urgent, some of them through the emergency pathways for awards that I think you're all aware of, and show that indeed the tissue chips can help identify existing drugs that might be repurposed for pandemic viral infections. Next one. And this led to a broader effort, which has a potentially unpronounceable acronym, speaking of acronyms, but what's that supposed to be is MPS Core. That's Microphysiological Systems COVID-19 Research Working Group. But this is a partnership among actually international agencies to bring together all of their MPS efforts and have members from, as you can see there, pharma, the MPS developers who are somewhat, they're both academic and companies, as well as regulatory agencies, et cetera. And again, this is something, and they all share data. And this is, again, something that would have taken, well, it might not even be possible, had been possible, but it was possible here. So it's not only within NIH, but much more broadly. So next one. In the clinical trial domain, I must say your institutions have just shined here in ways that really are inspiring. And the CTSA hubs being right in the middle of this, that we very quickly, in parallel with all the work that they were doing on N3C, on the database, turned to them to help answer two critical questions when it comes to clinical treatment of COVID-19. One is, would it be possible to damp down the so-called cytokine storm that is responsible for a lot of the suffering and death in COVID? It's actually not the initial viral infection, it's the immune system overreaction that actually causes a lot of the damage and death. Could we get a study started rapidly to do that? And secondly, to answer this ongoing question, which is whether convalescent plasma from patients who have recovered from COVID-19 actually works to treat disease in newly diagnosed patients. If we go to the next one, the first one that the CTSA program was involved in, many other programs or trials done by other parts of NIH, but I'm not going to talk about those because they're not NCATS. But the same principles apply. This was a trial run through ACTIV. Again, what I mentioned before. It's a randomized master protocol adaptive trial. For those of you who aren't clinical trialologists, what that means is it's an innovative clinical trial design that has been around for a long time, but has not really been used all that broadly because of concern over, well, gosh, it's kind of risky and might take too long and all these kinds of things. And all parties came together and said, boy, we got to do this. You know, we don't have time to test one of these at a time. We got to test 3 at a time. So these are 3 therapeutic agents, which came from 3 different pharmaceutical companies. And it's being run through mainly through the CTSA program Trial Innovation Network, is supported by Operation Warp Speed and BARDA, and it's ongoing recruitment. Next one is the one on convalescent plasma that I mentioned. You go to the next trial, next slide. These again are there are 2 multi-site trials run through the CTSA hubs. You may know that in one of the more, in my mind, problematic episodes of this pandemic, convalescent plasma started to be used in a expanded access protocol very quickly, and we did not learn, and we never could have learned, or shouldn't have expected to learn from that approach. Does convalescent plasma actually work? We actually don't know, despite having over 100,000 people in this country treated, and that's because randomized controlled clinical trials were not done in the size and complexity that they needed to be done. And again, this is a lesson that I've taken from this, that we've learned over and over and over again, that science cannot stop in the middle of a pandemic. It's often said we don't have time for science, people are dying, but that is one of many penny wise and pound foolish arguments that has been made, and we learned our lesson here. I hope we learned our lesson as a community. But in any case, through the CTSAs and through Operation Warp Speed and through NCATS and through the academic community, we're well on our way to solving this problem. The CONTAIN trial based at NYU and Montefiore and 17 other sites, and Pass It On trial based at Vanderbilt and 19 other sites, through recruitment now. And we've worked directly with them, with both of these, with the FDA and with other agencies to get direct submission to the FDA at the same time, making sure that what FDA needs is what the trials are doing, and promoting harmonization of the assay titering, if you know what that is. If you don't, it doesn't matter, but it's just to say that make these comparable after we're done. So again, huge amount of collaboration among sites, and I could tell you stories that about what these academic sites did that I hate to be a little, maybe I'll be a little Pollyannish here. They would bring tears to your eyes, literally, what these places did. It is a story that has to be told, and I hope will be told once all this is over. Next one. The NCATS has always been big on community engagement. One of the reasons is that our definition of translation is that translation has to make it to health. That's what we're interested in. Translation goes to health. And where does health happen? Health does not happen in a lab. Health doesn't happen in a clinical trial. Health doesn't happen once FDA has proved something. Health happens in the community. And that is the only place where translation is completed. And sometimes we forget that. And patients don't forget it, let me tell you. And they know. And we have a lot of work to do to understand how to do this well. And so CTSA program has been doing this for many, many years. We have a lot of trusted community partners, which were brought into this very rapidly to address, among other things, the health disparities that I mentioned before. There's a whole site here on the NCATS website that we've created that I think you might find useful. It's linked there at the bottom. Go to the next one. And I just want to mention two programs you've probably heard about, one which the CTSAs have been involved in. Go to the next one. One is the SEAL program, and the other is the RADX program. These are SEAL run mainly by NHLBI and NIMHD, and RADX run mainly by NIBIB. But all of these were broad collaborative efforts to, in both cases, reduce health disparities. If you go to the next one, I want to make the point that these are the SEAL sites, and of the 11 sites, 8 of them were the community engagement groups at the CTSAs, at these institutions. And so my point here is that a lot of the expertise and community building, expertise building and infrastructure building, if you want to use that term, was brought to bear on this very, very rapidly through this program. If you go to the next one, the RADX program, I think you're aware of. This is the Rapid Acceleration of Diagnostics for COVID. I just want to make a point about the RADX UP program, which is up on the upper right corner here. This is to understand why it is that people of color and in rural communities and other places have had so much less testing, and how do we get diagnostic testing for COVID to those communities? Go to the next one. you'll see that, sorry, keep going. You'll see that a number of these sites, the RADx UP sites, underrepresented population sites, were also at the academic centers, which were CTSA supplement sites, or that is, there were CTSA sites that were RADx UP sites. So next one. I want to just finish up here by showing you some information which I think you'll find helpful about this ACTIV partnership. So what I showed you up until this point are some programs that were based at NCATS. Every one of those was a collaboration with at least one other institute at the NIH, and many academic institutions, and in some cases companies as well. And in each case, we made progress on sometimes kind of conventional outcomes, like what I showed you on the tissue chips program. That would be a kind of a common NIH outcome, but one that happened much more rapidly than normal. And then others, like the N3C program, or the SIRA survey, or the clinical trials, which would have been difficult or even impossible, and certainly impossible in the time that it took. And I know when I say time, I hope you're all thinking that the time is not only individual patients' lives, and that of course is who we report to, and that's what's most important, but the fact that these agreements, and they all have behind them agreements that many of you put together, it was done in sometimes 1/10 the time that it normally takes. And so I think we really need to ask ourselves, well, gosh, you know, we really got a lot done in a very short period of time. And we somehow, you know, the world didn't come to an end. As a matter of fact, it got better. So, you know, how do we keep doing this? So now I'm going to tell you a story about ACTIV, which is that same lesson, but expanded even a little bit broader. So the concept here was, you probably know that back in April, it was appreciated that, you know, NIH is an important piece of this, but there are also other federal agencies and lots of companies working on COVID therapeutics and vaccines. And so shouldn't we have a nationwide coordinated response to prioritize and test therapeutics and vaccines? And virtually, well, most of the therapeutics and vaccines that you've seen and heard about have come through this program. They may not have talked about ACTIV, but they've come through this program. So next one. So I want to show you this, and normally I don't show this, but for this group, I think you'll appreciate it. If you can imagine the complexity of putting together an agreement to have all of these organizations come together, this would have, and the FNIH ran this, this normally would have taken 2 to 3 years to put together. It got put together in 2 weeks. 2 weeks to get agreement from all of these. And if you go to the next one, I'm going to give you a little shock and awe here. This is what the executive committee looks like, and then a leadership team, which has about 200 people on it. It's NIH, it's pharma, it's Operation Warp Speed, it's FDA, it's BARDA, and then there are all these working groups. And if you go to the next one, this is actually what it looked like, because we had to work very closely with Operation Warp Speed. That's where a lot of the money was coming from, but they were also working on diagnostics and vaccines and therapeutics, and as we were as well. And this had to be coordinated in real time. So how do you do that? It required an enormous amount of cooperation and getting to yes very, very quickly. And it happened. So again, why did it happen? So I think we have to continue asking ourselves that, and don't we want to have this continue happening, so that we can all spend more of our time doing important work and less time, you know, doing agreements and, you know, administrative burdens and all that stuff. So next one. I just want to give you a little bit of a sense of how this worked, because I think it's important to, you know, you're going to think about this from, as you should, from an agreement standpoint, but to just show you how complex this was, that there were 4 working groups, which were public and private, looking at preclinical assets, mainly drugs that might need to be developed a little bit more. There was a platform for mainly academics, but also companies to submit ideas, and then those went into a prioritization funnel within ACTIV, and then when there were preclinical assets that were viewed as being promising, they were assigned resources. Same thing happened in the clinical space to look at hundreds of molecules, drugs, which were ready to go into the clinic. Which ones should we prioritize? And out of that is one of the immunomodulator protocols that NCATS is running. Now, one of the things that many of us have wanted to do at the NIH for years, but we've never been able to do, is to have a snapshot of what is all of the clinical trial capacity that all of the institutes and centers have, to have it all in one place. And we realized, well, gosh, we can't build a new clinical trial network to do this. We got to use the capacity we already have. And of course, NIH has many, 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 but we didn't know across the NIH. We'd never done this before. It's remarkable because we never had a reason to. But gosh, when we put it together, the richness of mainly the academic clinical trial capacity in this country is extraordinary. But it had not been mapped or coordinated. And then, of course, there was all the development of vaccines, which was analogous to the therapeutics. So next one. I just got to very quickly, don't worry about the details here, but what I want you to get a sense of is how much work was done in an extremely short period of time, both with preclinical testing, with this prioritization I mentioned, a national strategy for non-human primate research using taking advantage of the primate research centers at a lot of your institutions, as well as the small animal testing facilities that are all over the country. We clearly had to utilize those limited resources in an unprecedentedly effective way. So how do we do that? We've never done that before either. Creating a public database that I mentioned, the Open Data Portal, to share all this this data, and then creating something which will be near and dear to your heart, conducting a matchmaking process to pair promising compounds with preclinical resources in the academic world, and with funding for Operation Warp Speed or others on an ongoing recursive basis. Next one. And you've seen the Open Data Portal. I showed you this before. This is the animal model one for small and large animals. Next one. The clinical therapeutics working groups, same idea, prioritizing agents, and then figuring out where to test them. In this case, they're not at small and large animal testing facilities, they're in humans. So then one had to, of course, go to clinical trial networks, which had been defined by that exercise that I showed you before, the clinical trial networks. And if you go to the next one, this is how it was done. Taking publicly available data, submission from investigators, surveys that have been put out, clinical agents, and many different kinds of mechanism, direct antivirals, immunomodulators, antibodies, et cetera. These were all scored and then had resources assigned based on the basis of that nationwide prioritization. It's one of the things that was kind of painful for me, I must say, listening to some of the commentary nationwide, a lot of which was justified, but there was a lot of talk about how there was no coordination among the federal agencies or with the academic centers, and there certainly could have been more, of course, but there was an enormous amount going on already, and I must say, if there was one thing that lagged here, it was the communications, which I must say was actually, in my own view, lagged and was quite poor, and it was done because of concerns about getting out ahead of one party or another and keeping everybody in the tent, and I think we can appreciate that, but it led to a misperception that I think still goes on to this day, perhaps. So next one. The clinical trial capacity working group I mentioned before. These were not only academic sites, but CROs as well. This was not only US, but international as well, because a lot of these turned out to be international sites as well. And we all have that, and we have this in this amazing dashboard now of what capacity exists nationwide, which we never had before. Next one. So this is the portfolio of clinical trial protocols. There's 5 of them now. The one that I mentioned before, ACTIV-1, this is run through NCATS, but it's these 3 immune modulators for cytokine storm. ACTIV-2 and 3 are on neutralizing monoclonal antibodies in outpatients and inpatients. ACTIV-4 is on anticoagulants because we got to appreciate early on that vascular injuries, stroke, MI, other things were very common in these patients. And then ACTIV-5 being a phase 2 study, and all of these done in an unprecedented and collaborative way. So next one. So if you want more information on ACTIV, I'm not going to go on anymore about this, but there is a lot to learn from this about how we collaborate with each other and how effective it is when we do that. So I think the last slide is some sort of takeaways from my point of view, and everything I've shown you here, and pretty much everything I've said with a few exceptions, has been data, not opinion. This is opinion, but I think we're at that part in our presentation, and I hope it will lead to a lively discussion. So I just click forward once. My takeaway here is that it is possible to do translation, that's what I've been describing, much more rapidly, to go from fundamental discovery to therapeutics and vaccines much more quickly than has historically occurred. And you'll just reflect for a minute, when the public first heard that these therapeutics and vaccines had been developed in such a rapid time, their immediate reaction is there must have been scientific corners cut, they must be unsafe. And I could tell you that is not the case. I was in this from day one, and there were no corners cut. The reason is a lot of the operational inefficiency, bureaucracy, duplication, lack of sharing, all that part, the science problems remained. The operational problems, to a great degree, evaporated. And it shows you that the work you are doing in the FDP is critically important, because we got therapeutics and vaccines to people in an unprecedented time by doing exactly what you're trying to do, but it was pushed by this public health emergency. So next one. So again, in my opinion, why did this happen? This is the first time in my professional or personal lifetime, for that matter, that the feeling of urgency, which I feel, and many patients with untreatable or poorly treatable, potentially lethal diseases feel every single day. Think about a cancer patient who's been told, well, maybe we don't really have a treatment for you in a year, maybe, or a rare disease patient who, for 95% of those diseases have no treatment whatsoever. They feel urgency every day. The patients do, the parents do, the family members do. But for the most part, the rest of the participants in the research ecosystem who are not critically ill or don't have family members who are critically ill don't feel that urgency. Everyone felt that urgency and still does in this pandemic. And that urgency led to an almost immediate recalculation of benefit risk. That the risk was higher, therefore we are going to recalculate how much risk we are willing to take to our own organizations or our own careers based on that urgency. But again, patients feel every day, but most people have not. And what that led, this was really interesting for me to watch, is without my asking anybody, they spontaneously started to write me and ask how they can share their expertise, their data, et cetera, et cetera. So we're not pulling data out of people for the first time. They sent it to us. They wanted to share. They wanted to collaborate. So what that tells you is that people intuitively know that these are the behaviors that will lead to more efficiency. Collaboration, openness, sharing, all of those things are what people did automatically when this happened. So this led to proactive collaboration. among the public sector, it will shock you to learn that not all the ICs always work together. They can be, silos just as much as academic institutions or companies or anybody else. They all work together. Companies in the private sector, public-private working together from a desire to share because they intuitively knew if they did that, things would go faster. And in fact, they did. So the next one. So if we think about where we are now, and my heavens, I had hoped I could have talked in the past tense when it came to COVID. I can in no way do that now, unfortunately. But we will be to a point where we are able to think about this in the rearview mirror. But I want us to realize that this is a potentially, and that word potentially is really important, potentially positively reinforcing cycle, because the increased efficiencies that we saw and are seeing have led to increased productivity by whatever metric you have, publications, products, patents, whatever it is, increased productivity, which has led to increased return on investment despite sharing, right? So nobody wants to share because they'd rather have a whole loaf than no loaf at all. More often, there is no loaf. So there's nothing to take credit for, but people don't share in part for that reason. Here, what we found is that the productivity, just to take some numbers as an example, productivity goes up 4x. Partners have had to share, so they have to share, they get half as much credit, but they have 4 times as much to take credit for, therefore their productivity doubles. And that's what we saw over and over and over again. So in other words, the pie got bigger and there was much more credit to go around. So that is positively reinforcing. So the next one, our last one. I think we have to realize, and I know you're all aware of this, that many of the conditions, the exceptions to regulations or policy, a lot of the things that NIH did through OER, which were really quite Herculean, and a lot of things that you did, a lot of things that FDA did, a lot of things that CMS did, and all that additional funding are likely not going to continue. We are going to, human beings in our culture is remarkably homeostatic. We have an incredible ability to go back to what we did before, unless we all take proactive steps to maintain these. And so what we're trying to do, and what I'd urge you to do, is to try to put your finger on what the perverse incentives were and are, and what the policy issues are and were that are preventing this kind of incredible productivity from happening every day for every disease. And I'll just leave you with one provocative thought, which is, if we do not do this for every other disease, then we are implicitly saying that the lives of patients with cancer or rare disease or other terrible diseases that affect the human family, the lives of those people are less important than the lives of people with COVID. And I don't think any of us would say that, but I think we have to hold ourselves to that North Star, because those are the stakes. And we've now shown ourselves that in many cases, this is not a can't do it, right? I mean, we're all familiar with, you go to a partner and say, well, I'd love to do that, but I can't do it. They can't say that anymore because they just did it. It's not a can't do, it's a won't do. So the question is, how do we hold ourselves accountable as a community, both within our sectors and between them? So I want to thank you for your attention. I've tried to make this, both interesting and informative and a little provocative, that we might lead to some good discussion. So that's the end for me, and glad to engage in whatever discussion time we have.
Thank you, Dr. Austin. Michelle and I are going to tag team reading the questions. We have a good number of questions. I'm going to start from the beginning, going back to earlier parts of your presentation topics. The first question we have is, first of all, thanking you. This is wonderful. how do we make our ability to rapidly do this work in a sustainable way? And this is something that I think we've all talked about. Other non-COVID work has been impacted by this.
Yeah.
And there's a lot of mention about burnout while responding with the urgency. How do we build this to be a sustainable, transformative infrastructure beyond COVID?
Yeah. Boy, that's the $64,000 question, right? Because it's, as I often say, at NCATS, we have the privilege and the trouble of not being able to choose a disease we work on. We have all 7,000 diseases that affect the human family. And I often say that, the other 6,999 didn't take a vacation while we were dealing with COVID. And all those investigators who work on them got severely impacted in many cases. And we're still dealing with that. All of us are. I think first of all, I think we have to realize and hope and pray that a pandemic of this proportion will be, for all of us, I hope, a one-time event in our lives. So and the reason I say that is that the amount of oxygen, research oxygen, that COVID has sucked out of the room, I think is appropriate. It's proportionate to the public health crisis, the enormity of it. But happily, there are no other such threats around, and that's both a blessing and a curse. The reason I mention it is that there's nothing that makes me think that when this is all over, there will be something else that will occupy most of our time. So the question is, how do we deal with what in another setting would be the peace dividend of the increased efficiency, and apply that increased efficiency to many, 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 many other diseases, which added up would have the morbidity and mortality of COVID, but democratize that, I think, across all the diseases that we all work on. And I think that is the question that all of us have to deal with. I think that when I as you might imagine, I spent a lot of time talking to our investigators, grantees, et cetera, and I think what the ones who have been involved in COVID have appreciated that they have been able to do things that they would love to be able to do in their normal life, if you want to call it that, because nobody worked on COVID before, right? There were no COVIDologists. So everybody's going to go back to their day job. And I think the first thing we have to do, and what we're doing actually very, very actively, is to capture from the people on the ground what happened. And as a matter of fact, through the CTSA program, we're actually well on publishing a whole issue of the Journal of Clinical Translational Science from all the CTSA hubs. But what happened to the academic health institutions, and what was good, what was bad, what should we keep going, what should we stop? And the whole purpose of that is we want to apply that to all the other diseases that we work on. I think the other thing that we really got to focus on, and I might get in trouble for saying this, but what the heck, we're on the phone and we all agree with this. The public sometimes doesn't understand why biomedical research is so important. They don't understand. Most people, as you know, most people in the United States never heard of NIH. And so this is a teachable moment to say that your investment in biomedical research paid off. And wouldn't you want this to continue for all of your diseases and the entire research ecosystem? That is also something that I don't think we should let pass.
Thank you. And so I'm going to go a little bit out of order because this goes along the lines of what you just said. Someone asked, how do we foster a culture of urgency in scientific and administrative process responses to infectious diseases, moving from infectious to chronic diseases?
Yeah. in my experience, because I've thought a lot about that, there's two factors. One is to inculcate close and regular interaction with patients. You know, all of us lead very complex lives and we do complex things. And in our world, as well as others, it's really easy to forget, you know, why we're all doing this. And so NCATS has, in our organization, one of the first things I did when I became director was I said, every program we're going to do is going to involve patients as a member of the research team from the very beginning. And that, believe me, patients feel that urgency. And so even when patients aren't around, when we're trying to deal with something here at NCATS and we can't figure out what to do, I'll sometimes say to the folks that I'm meeting with, well, if you were a child, if you were a parent of a child with a disease like this, what would you want to have happen? What would you want the policy to be? And when we do that, a lot of the underbrush just goes away, and it becomes very clear what we should do. And what that leads to is a quite dramatic increase in job satisfaction, because people who had been separated from the ultimate outcome are now part of it. The second thing that I would mention, which is again kind of unconventional perhaps, is that we involve our administrators, our budget people, our HR people, the people who order reagents for the lab, our strategic alliances people, tech transfer, all those, they are members of that team. And I have made it clear to them that they need to do these three things too: collaborate, innovate, and accelerate. And that they are not in any way just, you know, a sideshow. They are the show. They are the engine room. And without them, nothing happens. And I've been amazed sometimes at how unusual that inclusion is in many of our organizations. You know, and I think some of it, you know, won't surprise you to learn that, you know, there's a fair amount of you know, prestige, you know, looking here, you know, and I can say this because, you know, the MDs get the highest prestige, and then the PhDs, and then you keep going down the list. And I don't think that way, and NCATS doesn't think that way. You know, a team requires every member, and so inclusion of, you know, for instance, just give you one example, our head of strategic alliances, and as Lily Portilla, some of you may know, is what we call tech transfer, reports directly to me. And that is unheard of at the NIH. But it is very clear that the agreements that she does and her group does are absolutely critical to our making any progress. So some of it's a cultural thing as well.
Yeah, actually, that was one of our first questions we received. Assuming this is a professor, researcher at an institution, they hit bottlenecks while everybody seemed, you know, they were all on board. They continued to, and they combined resources quickly to, you know, work on COVID. But this individual still continued to hit bottlenecks of administrative policy. So, you know, data sharing agreements, IP, IRB, ICOOK turnarounds, contracts with private partners. And so they're asking about pearls of wisdom, and I think you essentially answered it. It's a cultural, but if you have any other additional thoughts about that.
Well, this is a question that hits home for me because we're dealing with this currently in one of our clinical trials where the startup time for some of the sites has been remarkably slow. And it's, at first I thought, well, they're just overwhelmed with patients, but of course they're not the people taking care of the patients. it was because of a return to business as usual. And so I've actually been getting on the phone with all of these institutions myself and exhorting them to overcome their caution, I guess is the way to put it. And what I've seen, and depending on what the institution is, that helps to a great degree. You know, I've done that. You might know that actually Francis Collins has done that several times with our investigators and others. Janet Woodcock has done the same thing as head of therapeutics for Operation Warp Speed. So some of it is the general talking to the troops, if you will, motivation. But part of it is also buy-in from the next level up, and that's really what I think my involvement or Francis's involvement or Janet's involvement has given, that the person doing these agreements is worried about what their management is going to say, and it's always easier to do the same old thing. If you take a risk and something bad happens, you yourself are at risk, and so it doesn't tend to happen. So it requires cover from somebody higher in the organization. And I think one of the things that one of the things you should know that we're very actively talking about here at NIH is how do we as an organization incentivize our funded organizations to act in the way that we want them to, because we realize that, we've seen the enemy and he is us, right? I mean, a lot of the problems comes from NIH. So it is true, and it illustrates what I was saying before, that as soon as you take your foot off the gas pedal, a very large number of people will go back to their comfort zone. And I certainly don't understand completely how we keep people out of their comfort zone, but that's what we got to do until it becomes the new normal.
Okay, yeah, and we have a few questions along those lines. I want to go back to a question regarding, from a professor, a major difference between COVID-19 effort and past research efforts is that it was a more targeted approach to a significant problem, somewhat away from investigator-initiated research. Do we need to think about changing a portion of our conduct of research model and funding to directing research efforts to specific societal problems and away from pursuing investigator?
Yeah, it's a question near and dear to my heart, and does some people in the NIH ecosystem, that's a thermonuclear question, but I think it's a really, really important societal question that we as the community have an obligation to ask ourselves. And if we don't ask it, it's going to get asked for us. So I don't think we can play ostrich here. It will not surprise you that as the head of the NIH's Translational Science Institute, I think more resources need to be directed and more attention need to be directed in that direction. And it's not because of my desire to build my empire. It's because I started out as I'm a very basic science geneticist. That's where I got my start, and I've gotten to appreciate that the social contract that NIH made with the public at the end of World War II, we have largely fulfilled. And what do I mean by that? Going back to Vannevar Bush, the social contract was if you fund fund us to do fundamental research, we will understand the nature of health and disease as a basis on which to intervene and treat that disease. Now, we certainly don't understand everything about our diseases, for heaven's sake, but we do know enough to rapidly shift to more patient-focused outcomes. And what I've seen is, again, speaking from my experience, institutions are slow to change, and they're, NCATS is 2% of the budget. So, most of it remains, fundamental research, which is wonderful and necessary and all that stuff. So I wouldn't cut any of that by any means. But I do think, just speaking for myself, that I think we have an obligation to the public to deliver on the promise of that science for people in need. I used this number before, 95% of diseases which affect the human family have no FDA-approved treatment at all. We understand the molecular basis of almost every one of them. So I think that creates for patients suffering with those diseases, this creates a COVID situation. Why isn't more being done to take advantage of this? So I think it's a difficult question, because there's a lot of appropriate resources invested in fundamental investigation. And again, that's where I started, and I certainly don't, that's the seed corn on which translation is based. But I do personally, I think we have an obligation to do better there.
Thank you. We have a few questions around the general theme of one of our biggest administrative challenges is the inconsistent practices of federal agencies. Do you think this experience will influence a push towards centralization or other solutions?
Yeah, sorry for laughing there. It was as you go. So you struck a chord. Yeah. As you can probably tell from the conversation we've had for the last hour, I am an inveterate optimist, and I also like to think that when we demonstrate that we can do something and it has a good outcome, that we'll do more of it. So as a result, I do like to think, and I do think that that this effort to coordinate federal agencies better will continue. I mean, I don't know if you remember that ACTIV slide. I ran through it pretty quickly, but agencies which barely knew about each other, but they're funding agencies that fund all, BARDA and DARPA and DOD and NIH and NSF and all of these places. They work really independently, but now they all know each other. And so, I would certainly hope that in the same way as we practice what we preach, put it that way. That is, if we are going to ask you all to streamline and work together and come up with uniform practices, I hope that we will not be so disingenuous to not do it ourselves. I know, people like Jody, now that she's moved on to a new position, but and Mike Lauer, they are totally devoted to this. And I'm hoping that the other agencies will see so as well.
Great. That's good news for all of us.
Yeah.
But much of what we focus
you got to
on
realize
here,
some of this is purely self-serving, right? I mean, if we get this to work better, it's less work for us. So that's what we're trying to get people to realize. It's sometimes hard though, as you know.
No. So we're running out of time. We're getting a lot of great questions, and I just want everybody to know that if we're not able to answer, that we will follow up with Dr. Austin and be sure to post them. But there's another theme around how does your presentation and how these activities focused towards COVID could possibly fundamentally change how research is done in the new norm.
Well, I think one area that I hope will change, and it's already beginning to change, and being de-risked, though we still have work to do, is in the area of data sharing. So if we think about what our mothers all told us, many hands make light work, or a team is able to get a team with diverse players is able to do things that a team with all the same players can't do. Data sharing is probably the simplest collaborative mechanism we have, because you don't even have to know who your collaborator is. You just put out the data and they're collaborating on your data without you're even knowing who they are. Now, and what's been limiting to that is, of course, policies and a place to put the data. And you saw the new data sharing policy that came out from NIH a couple months ago tries to address some of those issues. But of course, the big elephant in the room is credit. The reason that people haven't shared is that you got to get credit for what you do. But I think what we've seen in COVID is a willingness to share that we haven't seen before, and therefore progress that we haven't seen before, both because it led to connection of puzzle pieces, which otherwise might have to wait for a publication and maybe not even then, as well as the fact that it allowed people to not do to not do experiments that they might have otherwise done. So there's a lot of saving of time and resources. But what I continue to be troubled by, and we really need ways to account for this, and we're doing it within N3C, is we need a community agreement. And I don't know whether it's possible to put this in statute or not, or whether it just has to be a community norm across all of biomedical research, that you don't publish on somebody else's data until they published on it. And I saw this happen. I spent 10 years at the Genome Institute before I came here. And of course, a lot of you probably know that that culture developed within the Genome Institute, and it worked. But the reason it worked is because it was a very small community, especially back then. We've tried to enforce that within N3C. That's 1,000 investigators across the research spectrum. We'll see how that works out, establishing community norms to give credit for data. I still think we have work to do on micropublications and getting credit for data sets, even whether they're not connected with a publication or not, and have those count toward promotion and tenure. I just think unless we do that, we're going to be asking people to act against their own self-interest. And, so am I going to get tenure or share my data? They're going to choose the first. They got bills to pay, they got a job to keep. And so I think the job of those of us in management and administration is to not have our researchers have to make that decision. And that's our job, not theirs. But we need to have that, continue to have that conversation, but I do think data sharing is an obvious route. I think the other thing that NIH, I'm hoping will happen as a result of this, which has already started to happen, but not as much as I would like to see, is having mechanisms, funding mechanisms that continue to provide alternatives to the conventional RO1 PI, one PI model, which just is wonderful for certain applications, but doesn't work at all for translation and doesn't work for a lot of research. And so we're moving in that direction, and I think COVID will accelerate that.
Well, thank you. I think one more quick question regarding, the benefits of the effort that you've described, but recognizing costs, both explicit and implicit, opportunity costs, research foregone. Any thoughts about these costs and how to manage both the explicit and the implicit costs associated?
I mean, the loss of ability to be productive, whereas the cost to many institutions have continued, and so there's a deficit there. Exactly. Yeah. As you know, we were hoping to get relief from the Congress. I don't think that's going to happen in any meaningful way. So what's going on now very actively among the institute directors is how do we how do we help our grantees and grantee institutions in any way that we can with the next year's appropriation or this year's appropriation, with the knowledge that anything we do to help institutions this year will come out of our ability to fund new awards. So, right, it's a zero-sum game. So if we try to, fill in a hole from last year, then we can't fund something this year. I think where all of us have come down so far is that we have got to help trainees first. That they are the ones in the early, the KTs, et cetera, the NRSA awardees. Those we have an obligation. Those people we have an obligation to, and so if they need to stay on another year, we got to give them another year, and it's going to come at the expense of other things we have to do. But I think we have an obligation to do that. And we're trying to do that across. We were talking last week actually about how do we have a uniform policy or practice across the NIH, so it doesn't matter what institute you come from, that happens. It's certainly what we're doing with an NCATS. I think in the absence of additional funding, I frankly don't see a lot of additional relief coming, because the Congress has said, and we can understand this, they have a lot of priorities, and everybody suffered, and you have your next year appropriation. If you want to, take this is not, don't take this seriously, but as an extreme, if you want to say, okay, we're just not going to fund any new awards in 21, in order to make people whole from 20, you're at liberty to do that. But of course, we're not going to do that. But that's the kind of Sophie's choice decision we're having to make beyond the trainees, which I think everybody agrees we've got to do.
Okay, well, on that high note, we should have ended it with some
sobering note. Yes, well, I mean, we're all.
But thank you.
Yeah. I wish that was a better. I wish that was a better answer. A lot of us and you all have been.
Right.
And who knows? We got a new administration. Maybe there'll be another. Right. There's already talk about another supplemental. So never say never. And believe me, we have not given up on this by any means.
That's great.
This current state of play. That's the current play from Mudville.
Okay. Well, thank you very much. That makes me feel better. And Michelle and I would also just like to thank you for taking the time. This is a fascinating and really informative and inspiring. It's very well pointed around areas that FDP is dedicating our time on, trying to, improve smooth research and reduce administrative burden for the research. So thank you.
Well, we need you all. You all are really essential. You're out there fighting the dragon every day, and that dragon likes to get bigger and bigger and bigger. So we got to keep fighting it, or else it is going to take over everything. So thank you.
Thank you so much.